Hey, Bio One students, let's kind of wrap up the rest of these macromolecule notes. I want to go over nucleic acids today, and I want to go over proteins, which is a big one. And that way you can fill in your rest of your foldable. Hopefully, you've been making. There's instructions on Google Classroom. Please get that done for me because we're going to take a test on this stuff next week. So, nucleic acids, I'm not going to spend a ton of time on, mainly because I got those, and I'll go over them in detail a couple chapters from now. So, just a little brief stuff about them, just a couple slides. They transmit and store genetic information. We're talking about DNA and RNA. Like other macromolecules, they have hydrogen, oxygen, carbon, the big three. They also have nitrogen and phosphorus, a couple that we've added to it. And they're formed the same way as carbs and proteins. Monomers, individual units coming together and making a big molecule called DNA and RNA. That we have a special name for their monomers. They're called nucleotides. So make sure you highlight what's in blue. This will be, I'll put the notes on Google Classroom today as well. Okay, so that's pretty much what nucleic acids, what they look like is they're made out of this 3-unit phosphate, a 5-carbon sugar, or either deoxyribose or ribose, depending on if we're talking about DNA, RNA, and one of the 5 bases. I don't know if you remember that from middle school, but we got adenine, guanine, cytosine, thymine, and uracil. So that's the bases. So that can be interchanged here. And there's basically 2 types: the RNA, which has a sugar ribose, and DNA, which has a sugar deoxyribose. Okay, so that's the 2 kinds. I'm not going to go in a lot of detail about nucleic acids because, again, we'll go over that in genetics. But I want you to kind of know that they're made up of this 3-part unit called a nucleotide. And if we put a bunch of those together, we get a huge DNA RNA structure. All right. So that's basically the nucleic acids in a nutshell. So pretty quick. Okay. I want to spend most of my time today talking about proteins. Okay. The fourth and last of the macromolecules. Same deal: carbon, hydrogen, oxygen. We do have nitrogen in there as well. Proteins are polymers made out of amino acids. So amino acids are your monomers. Okay. Look at all that proteins do here. Let's highlight these. They control the rate of reactions. They regulate cell processes, their cell structures. They transport. They help fight disease. They do everything. Okay. You may have heard that saying, you are what you eat. A lot of truth with that because proteins are made out of these amino acids. We break down foods in our digestive system into these amino acids. We reassemble amino acids into, blood cells, skin cells, muscle cells, and make structure out of it. So proteins do not just all the structure in our body, they're very integral in the functioning part of our body as well. Okay. Now, here's where it gets kind of interesting. So I'll give you a little bit of an analogy and hopefully it makes sense. So how proteins work, it's all about shape, very similar to talking about the lipids. It's all about shape, okay? So let me give you a simple illustration. I think this will make sense. There's only 20 amino acids, but we have millions of different proteins. So how can we make all these proteins, one of the most diverse molecules on the planet? How do we make that many combinations with only 20 moving parts, basically? Okay. And it's all got to do with an R group, what we attach. Okay. So if you look at a protein, let's look at the diagram down below. Okay. If you look at them, they all have this yellow part, the H, N, and H, and they all have this O, C, O, H. So the yellow and the blue part does not change. What changes is this stuff in the middle, and that's what makes different proteins. So let me give you a simple illustration. I have a great big tractor at home I help my uncle farm with. That tractor never changes. I hook a disc to it and I disc. I take off the disc, I hook a plow. I hook a hay mower. I hook a baler. I change my attachments. Tractor's the same tractor, but I can do multiple things with that tractor by putting a different attachment on the back of it, if that makes sense. I think that's probably the simplest illustration. So amino acids, they have these R groups that never change. That's the tractor. But what does change is the side chains, the attachments. Okay. And by doing that, the sky's the limit. How many different attachments can I attach? There's a lot of them. The same way with amino acids. So I don't change the I don't change the middle part of it. I change the side chains, the R groups. Okay. So the tractor stays the same. We just keep changing the attachments. Hopefully that makes sense. And then we get all kinds of different shapes. Okay. You got 4 levels. You got primary, which is basically just putting amino acids in a train car, hook them one after another, and the sequencing will change, and that will change the will change the look of that protein. Okay, so that's called primary, just straight lined, putting amino acids in order. Okay, then I can take it and I can fold it up. So let me show you an illustration. This is a very simple illustration, but I hope you get the drift. So I can have a piece of paper. That's the primary structure. Then I can take that and I can fold that paper, right? Now I'm going to get a little bit of a different look to that paper. So by folding it, I'm going to get all kinds of different looks to that same piece of paper. So that would be secondary structure, sort of like how we made our foldable, right? So now I got all these different folds in. I've changed my piece of paper, okay? Then I can take it and I can, this looks kind of crazy, I can make it a 3-dimensional shape, right? So I can wad it up and make it into that kind of shape, or that's tertiary, okay? Or I could take a bunch of them and wad them up, right? And then attach all of them together, and I get this crazy looking immense structure with all these different wads all wadded together. That would be a quaternary structure, okay? So hemoglobin, if you look at hemoglobin up here, that would be a quaternary structure protein, okay? So they can be very simple, just a chain of amino acids, or they can have folds, they can have, wads or tertiary 3D shape, or they can have a bunch of them hooked together like you see up here. We'll highlight it here with the hemoglobin. Your blood looks like that, okay? So kind of crazy, okay? Funny video. You can watch it when I post the notes after today. I'm not going to show it to you now because it echoes so bad on my computer, but you can watch that. Here's a good picture of quaternary structure. You can see hemoglobin. That's what it actually looks like. And here's the different looking moving parts. Okay, so we can get pretty complex with some of those things. Okay, I think this is kind of a funny picture too. This is your spider on drugs, but sometimes drugs like LSD and caffeine and hashish, it can change the physical structure of whatever you're building, in this case, spider webs, okay? So that's pretty much the macromolecules. So what you can do is take what I have in blue and red. Again, I'll post these notes online, and you guys can put that in your foldable, and you can use that foldable on your test, okay? Hey, that's it for 2.3. We'll do a little bit on 2.4 next week, and then we'll take our test at the end of the week, all right? Hey, have a great weekend. Have a good day, and we'll talk to you later. See ya. Bye.